Okay, thank you. So my name is Tim Bromage. I'm in the Faculty of Dentistry at New York University in a department of biomaterials and biomimetics, probably a category of person you've never encountered at such a brown bag seminar before. Invited here by Igor Smolyar at NOAA to give a presentation about the work that we've been doing concerning the testing of environmental water. And the title is meant to be a little provocative, you know, how to inventory the environmental waters of the world one atom at a time. Of course, that's crazy, but that is exactly the crazy occupation we've decided to have for ourselves. And I'll explain what that really means, which is that we intend on a global scale to evaluate environmental waters for the entire relevant inorganic spectrum of elements of the periodic table. Now, by relevant, what is meant is that there are some gases that require special technologies that we cannot account for, as well as some radioactive elements that we do not have permits for. So in the end, we have instrumentation for measuring 71 of the 92 elements of the classic periodic table, from lithium to uranium, that we measure in absolute concentration values, which is something you will never have heard before, because in fact our laboratory at present is the only laboratory in the world with that capability, to take an aqueous sample and to be able to select a fresh or saltwater sample from anywhere on Earth and evaluate the entire inorganic spectrum of elements. One machine, one lab. And that happens to be at NYU College of Dentistry. The connection to dentistry is a little circumspect, but I'll explain what that is in a few minutes. But this is exactly what we have in mind to do. Most of that table that you see there, we can access at concentration levels of parts per trillion, billion, and million. So ultra-trace, trace, and also minor element concentrations of elements. So this is the machine. I need to click the video only for you to observe something, which is the lighting of the plasma. I think it's coming, Erin. I didn't do that properly. Okay. Better with the technology than I am. Okay, so the plasma has just lit, and what that means is that an area, a very small volumetric area behind that plasma, has risen to an extremely high temperature, and the glassware to the left of that plasma is introducing a mist of any solution that we might want to dispense into the machine. I think from there, Erin, we can cancel out, and I promise there are no more movies. Okay, so the question is, you know, how hot is that plasma? And you could say to yourself, well, how about as hot as the surface of the sun? And you would have to say, not hot enough. Surface sun temperatures are around 5,500 degrees Fahrenheit. That's not nearly hot enough. Because in fact, the plasma generated that is necessary is in the vicinity of 7,000 degrees Fahrenheit. And the reason for that, something we should have checked, Erin, before we started, were those two internet links. Like maybe if you just say yes, it will actually happen. Did we have to accept? Yes, thank you so much. Okay, I'm a little distant from the microphone, but observe this. So when the element is a solid, it's appearing black and as a liquid and a gas. And as we increase the temperature here, you see elements slowly going out of their solid phase into liquid or gas. And it is only carbon left, just right at there we go, right at around, 6,460 degrees Fahrenheit, carbon finally goes into a liquid phase. So give it a few hundred more degrees, and virtually the entire periodic table then becomes volatilized in a plasma at that temperature. That's what that is showing. And then back to our presentation here. Okay. That was all that was meant to demonstrate. Okay, here we go. So the first thing we thought we ought to do would be to look at bottled waters, because many of them are sourced from springs, wells, and other freshwater sources around the world, as a way of actually doing what we call global ecological stoichiometry. You select the water from anywhere on Earth, and you look at its elemental properties for understanding the concentration and distribution of elements in the environment, which is ecological stoichiometry. So we did that, and these are results color-coded. In blue, these are ultra-trace concentrations in parts per trillion, and then parts per billion and million in the logarithmic scale on the right, which is yellow to red. And through this variety of about 30 odd bottled waters or so, which have been tested, we looked at lots of things. Here's one. So there are a number of bottled waters in which the uranium is sitting right at around the EPA limit, which I find fantastic, actually. Barium, consistently finding bottled waters which are hovering below the EPA limit for drinking water. Iodine, which is an unregulated element, but which consistently quite a few bottled waters are rather extreme in the amount of iodine which is actually being consumed. Arsenic, way over in a number of bottled waters. What's the EPA doing out there? What are they doing? Are they bothering to test the bottled water industry? I'm not sure. EPA limit for iron is 300. Consistently, the number of them are at 1,000, way over. Aluminum, oh my gosh, 50 to 200 parts per billion is the EPA limit of aluminum in drinking water, but we're consistently finding a number of them which are in the 1,000 parts. Sodium, it's just not enough heart disease around, I guess. Quite a lot of sodium. Even in bottled waters where there is claimed to be no sodium, if it's being contained in a glass, which is leaching sodium. So between production or bottling and consumption, an amount of sodium can actually enter into a glass contained bottled water. Lithium, totally unregulated. I can tell you, based on our numbers, that if you were to avidly drink these brands of bottled water, if you consume about 3 of them a day, you will reach your minimum therapeutic dose. In case you're just a little on the edge, psychologically.
Is that brand specific?
Yeah. Some of them actually boast the high lithium content. For those of you who are like maybe borderline, some kind of disorder, manic depression, then maybe you self-select this water and you don't even know it. But yeah, these have very high levels of lithium in them, and you can reach minimum therapeutic doses if you drink enough of it in a day. We find this incredible. We also find it interesting that so many elements of the periodic table are neither regulated nor monitored by the EPA. But that's not just the EPA, that's the world. So it's kind of interesting to look at these bottled waters. They pretty much, you know, despite all the claims in the literature, on the left has just come up some tap water in New Jersey, right? New Jersey doesn't, you know, have an especially wonderful reputation for its water, but it's just as good as any other water you can buy for $1 a bottle, pretty much, at least in terms of its elemental composition. And we also just at the end find it kind of funny that the claims that bottled water companies make, when, at least in terms of their value in respect of the periodic table, they're pretty much the same. But never mind. Where do we go from here? To perform a kind of ecological stoichiometry. What can we say about the environment because of having a particular sample of water? Now, these are the bottled waters, most popular brands of bottled waters, about 30 brands or so of them, which we've subjected to a statistical technique called hierarchical clustering, looking for patterns of element concentrations rising and falling together. And we can confidently observe from these samples of bottled waters those that are coming from volcanic regions, you know, springs emanating from areas that have a volcanic soil and rock, and those that are coming from limestone regions, which are more alkaline, by the elements that they contain. So already we can say at least something general about the environment just by observing how constellations of elements travel with one another in particular ground sources in freshwater. I'm only pointing out seltzers as a German bottled water because at first we didn't think that the technique was working because we had 2 seltzers water samples, and one of them fell into the volcanic and the other fell into the limestone rock region. And our German geochemist was a little upset by this and went back and looked into the geology of Germany, where seltzers has its springs, and they have 2 springs. And it turns out there's a geological fault right between them. They're only about 20 kilometers apart. And one of them is in volcanic rock and the other one is in limestone rock. So she was very happy after seeing that it actually, the system actually works. But the thing is, pretty much this instrument, you can put anything into it. On the left, there's tap water, well water, rainwater, lake water from lake, there's Lake Baikal there, river water, seawater, one from New Jersey and one from Barbados, and snow. Snow are the two columns on the right. And except for seawater, the snow that we collected was more enriched at higher concentrations with more elements than any other freshwater sample we've ever collected. And one of these samples is coming from high in the Italian Alps near the Austrian border at about 14,000 feet. And there, that sample has more elements than we've ever seen in a freshwater sample. Now, when we look at the wind, the prevailing winds that rise up and over the Alps, basically the snow is scrubbing the atmosphere, which is emerging from Western Europe, and enriching the snow in very high concentrations of a lot of things. We find this such a fascinating aspect of the research because ultimately, that snow melt enriches the Austrian and Italian rivers and lakes, and they maybe ought to be concerned about that someday. I'm not sure, but we find, but But what we find is that snow as a scrubber of the atmosphere of elements, because if you were to take the surface area of a snowflake, it's enormous. And on its floating journey to Earth is basically scrubbing the atmosphere, telling you about where that wind comes from. And we currently have a project in Mongolia, which is an enormous country from east to west and north to south, covering lots of different habitats. And we've collected snow samples from around that country, one of the largest countries on Earth, but which has snow pretty much everywhere except the Gobi Desert, in order to evaluate snow against the prevailing winds that come from like Kazakhstan and Russia and China. But as well, here's bottled water a couple of bottled waters. There's on the left, the first and second columns are bottled waters. The third and fourth, those are two wines. And look at the very bottom, the amount of uranium and lead in these wines is extraordinary. If you were to drink these particular wines habitually, you would become lead poisoned in 10 to 15 years. Yeah.
Can you take the actual bottle and smelt it and do this test without the wine or without the water? It's like, how do you know how much of this is the actual content versus contamination of the bottling process?
Yeah, we can do better than that. That's a great question. The question is, can we also evaluate the glass to determine if the glass is contributing? It definitely is. For instance, we tested Heineken beer, and the Heineken bottle is green, and there was an element that I found a little upregulated. I hadn't seen in other samples we've tested before, and I looked it up and discovered that it's an element added to glass to make it green. And so we know that glass is a major contributor to providing elements into a solution. In these cases, what we would do is we would take the bottle and we would laser ablate it. So next to the machine in which we're dispensing solutions, all we have to do, even while the plasma is lit, we can remove the glassware and put on a bit of glassware that leads directly through a tube to a laser ablation, 193 nanometer a laser ablation instrument, just next to this next to our elemental ICP-MS spectrometer, and evaluate a solid. So we can evaluate the solid directly just by laser ablating and looking at the particles coming from the laser, volatilizing them the same way that we volatilize the solutions. And in this case, because we've actually looked at more than 1,000 wines, and we've already done some analyses of more than 250 or around 250 or so, and it's very clearly a national issue. We find that these kinds of concentrations, just as for instance of the high lead, are restricted to certain countries. So it's in their processing that this lead is being introduced. Their infrastructure is adding lead. And in another country, it might be aluminum, which is way over the EPA limit by 5 or 6 times. And there, it is also an aspect of the wine processing technology of that country. So these problems tend to be limited to a particular way of producing wine. So it's all about the production. Although I will say that we did look at a cesium, a stable isotope of cesium, which is a byproduct of nuclear fission and a byproduct of nuclear power plants, which is emitted out the smokestacks with every other byproduct of operating a nuclear power plant. And we see a logarithmic increase in the amount of cesium the nearer a vineyard is to the nuclear power plant. So the grape is also telling us a very intricate tale about where that grape was grown. in addition to the processing. There's a lot that goes into it. Anyway, we found that, but we've also, there's some beer, middle right, and then to the far right, a couple of milk samples, which, if you'll notice, the third from the bottom is lead, which are right near the EPA limit for lead, which I find a little unnerving, because children are drinking more milk than anyone else, and they will become lead poisoned. And already infants are having their blood tested, and even as infants already finding that their lead levels are rising close to 5 parts per billion in their serum, which is considered lead poison.
Where was the milk sample? Is that U.S. or international? Or?
No, that's one New Jersey and one upstate New York.
Thank you.
I think it's a story, and we don't know. We have a grant to study it, and we've already collected the milk samples from across the entire east-west breadth of New York State, which is a really wide state, sampling air quality basically along that entire region. That these two particular dairy farms, they're about 100 miles apart, but they are in the wind stream of the air that rises up as a southwesterly, which any one of us looking at a typical weather map in the wintertime can tell that the industrialization, industrial activity in Pennsylvania and New Jersey is bringing lead, falling as snow or rain in New Jersey and in New York, and then that's fueling the grass and hay, and then the cows eat that and give the lead back. So that's a story. But we don't know that that's true. But then we can, in addition to that, we can squirt in just, blood is on the left, that's my blood if it has a T on it. Plasma, so the cells, fibrinogen have been removed. And then saliva, and we even peed in a cup in urine. And practically with no sample preparation, we dispense these solutions into the instrument and get a complete relevant inorganic spectrum from them. You'll see on my blood, plasma, and urine, or saliva rather, you'll see a yellow spot, which is my lead level. And I was clinically diagnosed as being lead poisoned after I saw this. I went to a clinic and they said, "You are lead poisoned. You are way over the normal limit." And so I found the source, which was an offending cup that had a glaze that I was using every single day of my life, at work and at home, a set of cups that was feeding me 3 times the EPA limit in every cup I had. So I became lead poisoned, and I had symptoms. It's originally why I went to the doctor, because I didn't know what it was, and pretty much confirmed that I was lead poisoned. The point is, from any aqueous sample on Earth, as you can see, from tap water to oceanic water to wine and milk and saliva, we can address the environment in which those materials are coming. So to do ecological stoichiometry properly, which has never been performed on the whole periodic table, not in the history of this field has anyone ever evaluated the entire periodic table of any place. So we went to a Central African lake habitat called Lake Chilwa in southern Malawi on the border with Mozambique. And at that place, we decided to actually perform such a study, and we're in the middle of it right now. So we collected lots of things. That mouse is just sleeping, by the way, so don't worry about it, right? So we collected water samples from the lake, from the rivers, samples of trees to get the annual rings, gastropods, lots of mammals, mice, pigs, And as you'll see in a moment, an individual, a fisherman living on that lake, whose corpse was donated to the medical school nearby this place in which we sampled the bone, in addition to monkeys and a number of other creatures from other classes of animals, monitor lizards, birds, toads, frogs, fish, etc. And having collected in that environment, so here's an initial result, just as an example, in which we collected upstream, say 20 kilometers from the lake, and sampled along the rivers to the lake. And the lake, incidentally, does not have it's the very last lake in the African Rift Valley, does not have an outflow. There's no river emanating from it. The only reasons that this lake maintains a more or less stable depth is either because of a runoff which is underground or from evaporation. So just you can even see in this pattern, these are alkaline earth metals, just taking them as a group, that the red, being strontium, is rather high near the cities and the villages, some 10, 20 kilometers away, but virtually disappears by the time it gets to the lake, all having been absorbed by biological activities, by animals actually taking in that strontium and using it in the production of its shells or its bones and whatnot. But we would never have anticipated the complexity of the elemental map, that there would be such a variability in the manner in which the elements change in their concentration from upstream to downstream into the lake. We would have assumed even that the lake would become super concentrated, having no outflow, but it isn't. So these kinds of patterns and we give potential clues as to the reasons why we see the elements we see at the top of this slide. The strontium we think is probably due to coal ash, which is one of the primary means of creating fires for warmth and for cooking in Central Africa, is coal. The calcium, which is in the green, is probably coming from calcium ammonium nitrate fertilizer, which is used by the farmers there. And barium, barium is mined not so far from this area, so there's relatively high amount of barium. And magnesium, which is an element elevated in calcareous soils, which these are. So we're beginning to understand the human element as well as the natural environment and how it works, how it isn't, this is not a linear map. This is a very kind of non-linear world that we're encountering. And this is only the alkaline earth metals. We haven't even begun to analyze all of the rest of the 60 some odd elements. So now, in the first slide, you may have noticed that I'm responsible for what's called the Hard Tissue Research Unit, and I have to apologize to all of my NOAA colleagues that you will now learn something about hard tissues. But please bear with the 4 or 5 slides of it, because it's coming back to the water. Because the hard tissues are going to tell us something about the water. Here is a tooth. It's a lower front tooth, incisor tooth, and the diagram to the right is a cross section of it, what it would look like. Teeth grow like onions, and they have daily formation events which can be observed and measured in a microscope, and they're also near weekly growth lines in enamel, which are called striae of Retzius. And there are these things called predicamenta. And if you, let's say, look tonight carefully at your front teeth, perhaps your upper front teeth, central incisor teeth, you will see these bands crossing the tooth. And they represent the near weekly increments of growth of that tooth when you were a child. They're visible by eye. So, and here's an image, an actual image of a cross-section of human enamel. You can easily see these near weekly events. It's not a mystery now what they are. These events actually are structured into the life history of many mammal species. These are lots of primates involved. I'm pictured in upper right because my tooth actually contributed to this research, and my rhythm is 8 days, courtesy of a lollipop lamb chop that I bit into a little too hard and broke it. And the dentist kindly gave it to me later, and I made a thin section and counted the number of days. I'm an 8-day person. Now, but that's fascinating in itself, because if you look at this chart, that this rhythm in the tooth is actually related to body mass, which means it's actually related to metabolism, which means that the tooth or bone structure, which I'm going to show you in a moment, is actually a reflection of how you process materials from the environment. At what rate do you do that? And that becomes important for ecological reasons. Now, it turns out that an archetypal unit of bone called the lamella is that thing called the stria of Retzius here. Oh, it only likes going back one slide. That's okay. It's not so important. You remember what that looks like. Oh, well, you can see the image on the left there, just reproduced. Those stria of Retzius, that length of time between 1 day and about 9 or 10 days there. That length of time between one stria of Retzius and the next is called the repeat interval, and that is actually the frame of time that it takes to form one lamella, which means if we have a tooth from an individual, we can then calibrate the bone. This rhythm, by the way, is called the Havers-Hallberg oscillation. It's a metabolic rhythm which has been observed in mammals as a many days rhythm. It's a fascinating area of research, but that's not the purpose at the moment. The purpose is our being able now to calibrate the bone and use it for saying something about the lake we were just observing. So, as it says on the left, this cross-section of the mid-shaft human thigh bone, the femur, from a 26-year-old fisherman who made his living on this lake. And if we just look at a little bit of that bone, you can see that we're stepping way back, but all those lineations over which this yellow arrow is strung are the lamellae. And every single one of them in this particular fisherman was also 8 days. Now, because we know a lot about this skeletal biology of the people from Central Africa, we can actually put these lamellae in real years. And courtesy of data that we have from meteorological services of Malawi, we can then look at aspects of this man's bone development and relate them to environmental phenomena. Remember the 8 days. And then this is, in fact, what Igor Smolyar has done with this data, in which we parsed out 3 years of those lamellae and looked at its structure. And in the very bottom, you can more or less visualize 3 humps, which is 3 years. So it would seem that something about the variability in thickness of these lamellae is telling us something about growth rate on an annual rhythm. And by the way, Central Africans, for the large part, are still subsistence farmers and fishermen, and they are subject to the waxing and waning of food supplies. And it could very well be that this seasonal signature is from when this person was growing up, he was susceptible to those fluctuations in food availability, and that maybe this is the reason for it. But just for the fun of it, on the right, saying, well, you know, can it be that the rainfall in those particular years, which was in 1994 to 96 or so, or 1993, 94, 95, these lamellae were being formed, and this is the rainfall variability in that year. Is it possible? Well, to ask that, we come back to the periodic table. Here, in the blue, we have an isotope ratio of zinc over strontium against the annual rainfall over the 15 years of the primary lamellae that were laid down when this boy was growing up between the years 1981 and 1995. And it kind of, you know, that we're not deep enough into the research to develop the tools for determining the significance of this, but by eye, we kind of begin to understand that isotope variability is in this man's bone, having used the laser ablation across all of these lamellae over all of those years, giving us an elemental signal, which is more or less consistent with variability in rainfall over those 15 years. So here's an example where we took the laser ablation of this instrument and laser bladed across all of the 15 years, and every so many seconds collected the isotope data. And then we did this relatively, in this case, zinc over strontium, and see a pattern that more or less indicates that the elements which were taken in to the bone structure and held there in this individual are mimicking something going on in the environment. So the idea, we're getting to the end, the idea here is that we finally are able to evaluate an environment in which we have, you know, modern animals and plants and soils, even crops. We sampled crops at Lake Chilliwack, and the water, to be able to understand the flow of all of the materials in that place using this instrumentation that we have, to know how that system works. And in our case, we were rather struck by our analysis of the fishermen, because basically, when we looked at the full periodic table, I only showed you zinc and strontium ratio, but the full periodic table of that fisherman, if you look at it, mirrored the data that we retrieved from the lake. In fact, we had two Excel files in front of us. One of them was the laser ablated data of the full periodic table from this man's bone. And on the other, we had the full periodic table in the elemental composition of the water from the lake. And they were the same. You would be hard pressed to know which table you were looking at. So this man was mirroring the elemental composition of the lake that he made his living on. And that aspect of this work means that we should now even be able to go to the fossil record. And this is actually a site that I helped to pioneer with my friend Friedemann Schrank at the Senckenberg Research Institute in Frankfurt, Germany, a site in Malawi, in northern Malawi, which is about between 2 and 4 million years of age. And we've recovered early humans of 2.4, 2.5 million years of age there. In fact, the oldest known representative of the genus Homo comes from this site. That we can now take our knowledge of how to piece an environment together from the ecological stoichiometry point of view, whole periodic table of animals, plants, soils, and water, and now translate that information to the fossil record and retrodict even the chemistry of Paleo Lake Malawi. Paleo Lake Malawi is the second deepest lake in the world, only second to Lake Baikal in Siberia. It's the third largest lake in Africa. It's a monumental lake, but it was there 2.5 million years ago. We should be able to now retrodict the chemistry of that lake 2.5 million years ago, and even then begin to understand why we have the certain animals that we also retrieved from this site as fossils. We should begin to understand all of those connections. How can these animals be here because of the kind of lake that was nearby? We fully intend on doing this. We have the samples actually ready for the laser ablation. And we just undertook a laser blading course last week in Italy in order to be able to start this project. So we're doing it, but we're not only doing it geographically, we're doing it in terms of global scales of fresh and salt water. We're also doing it in time. So in order to give it a kind of a Z dimension, a fourth dimension to the work, to evaluate this over million-year timescales. So yeah, we're going to do it. And one way to do it would be via a site like this. And Erin, if I just click it again, will it happen? Let's try. This is the last slide. And I say yes, of course. Oh, here we go. Yeah. I'm going to sit down just for a moment, a few feet away from the microphone. But this is not a public site, but it's a site we've built. It's not public because at the moment the machine has a headache. There's a part that's too hot. We're trying to cool it down. The company is sending us an AC unit. But after it's fixed, this site will go public. So, but you can even go to it now just for fun. It's just called stoiki.com. But just as a for instance, just to show you, so here are just samples we've collected in our travels, you know, from the west coast of the US to Melbourne and Sydney. You can see on this map, we've collected lots of things. Let's just go to the east coast here. I think there's even a sample here that I took from the Shenandoah River with Igor not too long ago. Let's have a look in here. But maybe, let's see, I'm also kind of interested in knowing, let's see. So this happens to be the home of a colleague. He just built his home. And now if we click on his results, so these are only EPA regulated elements. You can see there aren't so many of them. In fact, there's one more than the EPA actually gives, but we wanted the table to be symmetrical. And look, nearly everything, if it's green, it means, you know, we're good. This man has heart disease. He's been diagnosed with high blood pressure. And look at his tap water. It's 3 times the EPA limit in sodium. He had no clue. That's actually illegal. There it is. So he wonders why he's taking medication for high blood pressure. It could be because his water is quite high in sodium. At any rate, you can see the possibilities that if we were to continue this sampling, and in fact, there are many samples that have been tested which are not on this map. We have maybe a couple of dozen oceanic samples from the Andaman Sea to the Pacific. We have collected water. And at the moment, there is now a vessel which is traveling, circumnavigating the world, completely on renewable energy to highlight a carbon-free world. But also its mission is to collect microplastics. And at every location, over some hundreds of kilometers, navigating one of the oceans, at every sampling point for microplastics, they are collecting a sample for us so that we can look at variability across a stretch of longitude and potentially latitude, depending on on that particular leg of the journey that they're making. So at any rate, this technology is called inductively coupled plasma mass spectrometry, which is a very common technology, but ours is called simultaneous, which means it measures the whole periodic table at once, and at parts per trillion, billion, million levels of sensitivity. And as I said, there are actually a number of these simultaneous instruments in the world. There's 6 or 7 of them in the United States at the moment. But of the 30 or 35 or so in the world, ours is the only one that does this. And that's because we put in the effort to develop what's called the method to do it, which was definitely not trivial. It took 2 years to develop that method, something industry was not willing to do. Reason we were willing to do it because ours is the only one in an academic institution, and the cost benefit is not something we look for. We only spend money, we don't make money. So industry, they'll only evaluate the elements they're told they have to evaluate because of the costs. So at present we kind of hold a monopoly, but it's something that it's something that definitely, to perform ecological stoichiometry, not only one lab can do, but at the moment, if we receive samples from anywhere on Earth, we can do them. If you were to send your sample out to a laboratory, even a federal laboratory, for evaluating half a dozen elements, you would already be paying way more for the entire periodic table than we would have to charge. So for value for your money, you're getting all of the unknown unknowns now. You're getting all the stuff you don't know, you didn't know, in addition to the stuff you wanted to know. So that seems like maybe a silly distinction, but it isn't. Every single sample on Earth that we have looked at has always had a surprise. And that includes the oceans. And certainly the freshwater resources of this country. We have hundreds and hundreds of samples from national parks in the western and southern United States. And there are always surprises, especially with the snow that we collect there. So with that, I believe we have come to this point where I say thank you. And if you have any questions, I'd be very happy to address them. Yeah. The 4 people here are clapping if you can't hear them.
How do you take your samples in moving bodies of water, like the ocean or the river? Do you take it along the shore? Do you take it at the surface? Do you take it at the bottom at the dirt? Where do you take your samples from?
Yeah, so it's always critical to know exactly where you take them from. Oh, sorry. Yes, the question pertains to, at what depths, at what locations do you collect water samples in moving water? That is typically dependent on logistics. So, for instance, we have a proposal in to the United Nations to evaluate every river system and all outlets and oceanic water opposite the mouths of these rivers of Samoa. And some of these rivers, we can just wade into them, depending upon the season, and collect at any depth, but they're not going to be very deep. They're only going to be a foot or 2 deep. Others, we will only be just collecting from the edge, and we'll report that. And there are always GPS coordinates and notes on every sample taken. In the ocean, it's more difficult, but we have a technology for measuring at surface. So for the Samoa project, we'll be taking 3 samples at every location in the oceanic water off the coral reef. And that will be at surface, at 10 meters, and at 40 meters. and we just collect them at those depths. And then we just go around the island opposite every mouth of every river, 1 or 2 kilometers offshore, and collect the samples. So, but in moving water, it's just what you get. We have special tubes. If people collect water for us, they just can't put it in a jar and send it to us. We specially chemically prepare every tube that a water sample goes into so that it's chemically clean, so there are no ultra trace or trace quantities of elements that came from manufacturing. We eliminate those. We prepare the tubes and we then provide them to the people who collect. But you know, it's always just about logistics. And at this stage, we don't know what we're doing. We just collect. And then, you know, we'll make it up as we go along. No one's ever done this before, so we're kind of just in territory. We don't really understand the variability.
So the reason where I was coming from, like take New York, for example, where they used to have a lot of mercury in manufacturing. It's a heavier element. It's going to fall to the bottom. It's probably down in
sure.
the bottom of the rivers over there in the mouth. But if you're collecting from the surface because somebody doesn't want to drown, I get it. But you're reporting maybe higher sodium, but you're reporting mercury.
That's a great point. And we have considered this, and we've made one pilot investigation to date, understanding that phenomenon. Because Lake Malawi is the 2nd deepest lake in the world, what a great place to perform such an experiment. So Friedemann, who I mentioned earlier, designed a platform on which we can go out into that lake. And the lake has you know, the topology of the lake floor has been mapped. And we know where to go. And this platform was designed to go down the 750 meters deep that lake is. And we experimented with methods on collecting water at different depths already in that lake in order to look at a stratification of the elements in the lake according to their mass, just as you mentioned. That experiment was such a failure, I can't tell you, because we don't have any idea what we're doing, and nearly every single container I sent down exploded, or I should say imploded from the pressure. And so finally, I made a little hole in the cap so that at some pressure it would fill up. But I have no idea what depth. We collected the samples, but it doesn't mean anything. So I figured NOAA knows more about how to do that than I do, which is one of the reasons for my coming here today, because what an enormous opportunity. And even to perform such an experiment and then evaluate the life that lives at these different depths, which you can do. Lake Malawi sits in what's called a fault-bordered rift. And what that means is on the side where we are, on the Malawi side, rather than the Tanzania and Mozambique side to the east, on the west side of the lake, it's actually it's an incline. So you can, and we have a grabber on the platform, you can go down at any depth you want and grab what would be equivalent to a couple of liters of sediment and any life that's living in that sediment, and relate that life to that stratification of elements in the water. Why not? It's one of those questions you can just ask and then decide if it's something to carry on with. But we definitely want to head into that territory, but we need somebody who knows actually what they're doing in order to collect the samples at such extraordinary depths as that.
Well, it's just a beautiful place too.
Oh my God! I mean, I've spent decades there. So.
Because would you expect much? I mean, you're trying to compare these ancient bones to correlate them to the environment chemically. Would you expect much diagenesis over time? Much change in the, because like carbonates, I mean, this is apatite, but a lot of carbonates will tend to change over time as they get exposed to moisture and elements.
There's one of your big, I don't know what I don't know, but there are two answers.
Could you repeat the question?
Oh, sorry, I'm repeating the question again, which is about the diagenesis in translating this research to the fossil record and the bones and teeth there. Number one, if we perform this on enamel, it's veritably unchanged. So we're great there, but we don't know much about enamel yet. We're going to chase enamel up as a material for investigating, but it should be rich with the periodic table for sure. But many of the fossils are also bones, and we want to extract this information from bones also. Number one, we have collected the carbonates. We've already done all of the carbonate chemistry in the paleosoils of the site. Over the course of 1.5 million years, we have stratified carbonate chemistry already formed, already done. And this is a natural soil carbonate profile. And we see it oscillating with the known temperature gradients at that time. So we can correct for the carbonates to a certain extent. But in addition to that, a lot of the bone is still bone. We still have intact collagen, even in bone 2.5 million years old. We still have original hydroxyapatite. But of course, in laser blading, you can't decide, well, what's diagenic and what isn't. And we have to learn how to separate those signals. But because we have the carbonates, we have soil samples from where these fossils have been extracted, we should be able to say, here is our diagenetic signal, and here is our natural signal that's telling us about the environment at the time. We should be able to go to that place.
Maybe look for variations with depth into the bone.
Yeah. Oh, yeah. Absolutely.
More
and
effective, more
absolutely. And even if you make thin sections, you can often see that kind of tidal point of where diagenesis kind of got to and then didn't get any further. Sometimes you actually see that. Yeah.
And we did have some online questions.
Sure.
So someone wanted to know, after your analysis of drinking water, what is the safest option to drink?
Your tap water.
Okay.
Your tap water is probably safer than any unknown in a bottled water you might drink. I don't understand the point of it, actually. They all claim to be so different and full of this and full of that. Some of these bottled waters are so full of elements because they put them through filtering processes, even natural environmental water. They put them through such a stringent regime of filtering that they have to add elements back in because nobody thinks it's water they're drinking. It doesn't taste like water. They actually add stuff back in. And this is supposed to be a natural product. I mean, oh my gosh, 80% of bottled waters are originally derived from environmental water, but that doesn't mean there's a lot of processing involved. So for my money, I would trust the municipality more than I would a bottled water. Sorry, bottled water companies, but that's, after looking at all of them and looking at New Jersey tap water, why would, well, except for the fact that my friend has high sodium. Yeah, no, that's actually a problem. So, you know, I would say that if you can confirm that your tap water at least isn't over EPA limits in anything, then it's going to be just as good as any bottled water on earth. Yeah.
So is it higher sodium than in a soda can?
I don't know. I have no idea. We haven't tested any sodas.
So we are almost at time, so we're going to have to leave it there for today. We did have a few more online questions, so I will pass those on to you, Tim.
Sure.
And thank you so much for coming in today.
Sure thing. Thank you, Erin.